The forensic biology section performs tests to determine the presence of body fluids such as blood, semen, and saliva. These examinations are chemical tests that react with certain components of the body fluid. For blood, 2 tests are used. The item is typically swabbed to remove a portion of the stain or cut and placed in a testing tray. Once the chemicals are added, a color change will immediately occur if positive. For saliva, the testing is based on the presence of an enzyme or amylase. If positive, a blue color will develop in the testing tube. Semen testing requires a variety of tests. Typically, the item is viewed under an alternate light source to identify potential stains not visible to the naked eye. These stains are then tested for acid phosphatase. If positive, a glass slide is prepared, stained with 2 dyes, and microscopically searched for spermatozoa. Most sexual assault kits are now tested using a DNA process that only checks for the presence of male DNA. This method has drastically reduced the backlog in that area. If any of the serology tests are positive and the submitting agency requests it, nuclear DNA testing is then performed on the items. Some items are DNA tested that do not undergo serology testing, such as hair roots or ball caps. The testing involves cutting a portion of the evidence, extracting or removing the DNA from the item. Then a test is run to determine how much DNA is present. Next, polymerase chain reaction, or PCR, is used to copy the specific DNA regions that are to be examined. The DNA is then typed on a genetic analyzer. The result is an electropherogram which depicts the DNA typing result and looks similar to an EKG printout. This information is then interpreted by the scientists to determine if the known reference samples match or do not match the evidence samples. The foundation for DNA testing is no 2 individuals have the same DNA profile except for identical siblings. By comparing the DNA profile from the subject or victim to the profile from the evidence, a determination can be made as to did that body fluid come from a particular person. Based on a computer software program provided by the FBI, certain DNA profiles from evidence can be databased or stored. Depending on a state's law, convicted offender samples can also be stored. This system is called CODIS, for Combined DNA Index System. CODIS allows for searching of forensic samples to determine if certain cases are linked or were committed by the same individual, such as in serial rapes. It may also link unsolved cases to convicted offenders. In Georgia, all incarcerated convicted felons and certain probationers are collected for DNA testing to be searched in CODIS. The DNA profiles in CODIS are then uploaded to the FBI routinely for searching at the national level. CODIS searching is automatically done, and once a sample is in CODIS, it will continuously be searched on a routine basis. In Georgia, there have been over 1,000 matches since CODIS began, and matches will continue to increase as the database gets larger. Almost 250 Georgia offenders have been linked to crimes in other states.